Helpin Pharmaceuticals in association with Higher Secondary Principals Forum.
Hello students, welcome to the lecture series. I am Indira Narvekar from Govt Higher Secondary Sankhali, and I am here to take up class 12 biology chapter number 8, human health and diseases. Let us begin with taking an overview of what is health. Health is a state of complete physical and mental as well as social well-being. It is not merely absence of any disease or not merely absence of physical fitness. A healthy person has optimum physical fitness, mental maturity, social and economic well-being. A healthy person increases the productivity and thus contributes in the economic prosperity. To maintain a healthy lifestyle, one needs to have balanced diet, personal hygiene and regular exercise for mind as well as body, and thus one can maintain a very good health. Disease. Disease is any condition which will interfere with the normal structure and the function of the body. It is usually manifested through characteristic sets of signs and symptoms. Let us see how the disease can be classified. Disease generally can be classified into infectious and non-infectious type of diseases. Infectious diseases are the ones which are easily transmitted from one person to another. Examples of infectious diseases are AIDS, malaria, common cold, etc. And non-infectious diseases are the ones which will not be transmitted easily and thus remains restricted to one's body. So this type of diseases can be diabetes, arthritis and cancer. We all know that cancer is one of the major cause of death in India. The agents that are going to cause infection in humans are called as pathogens. A pathogen can be a bacteria, virus, fungus, protozoa or any other organism. Let us see which are the common diseases in humans. There are different organisms, as we know, which causes diseases, and these are called as pathogens. We will first do the bacterial diseases. The bacterial diseases are typhoid. The causal organism here in typhoid is Salmonella typhi. It is transmitted through contaminated food and water. Usually the target area for the bacteria to infest itself is the small intestine. The symptoms include high fever, stomach pain, weakness, constipation, headache, diarrhea causing dehydration. Usually the diagnosis of this disease or typhoid is done through a test which is called as the Vidal test. The next disease is pneumonia. The causal organism here is Streptococcus pneumoniae and Haemophilus influenzae. The mode of transmission here is basically the respiratory tract. It is usually the droplet infection. When usually the person sneezes or coughs, the droplets are transmitted into the air, and when a non-infected person or a healthy person tends to inhale these droplets, the healthy person gets infected. The symptoms of these are usually inflammation of the air sacs within the lungs, which are called as alveoli. There is congestion of the lungs with high fever, cough, headache, etc. Now due to infection, we have to also remember alveoli gets filled up with fluid and it causes severe respiratory problems. The diagnosis of this pneumonia can be usually done with X-rays or blood and sputum test. While other common examples of bacterial diseases are dysentery, plague, diphtheria, etc. Coming to the next group of diseases, viral diseases. As we know, the most common viral disease that we usually find is common cold. It is caused by a causal organism which is basically a group of viruses which are known as rhinoviruses. The target area here again is the respiratory tract and the respiratory passage. It affects the nose as well. The transmission again here is through droplet infection. When the infected person coughs or sneezes, the droplets are released into the atmosphere, and these when inhaled by a healthy person or when they are fallen on surfaces like the laptops, keyboards, pens, utensils, etc., and when they are touched by the healthy human, basically what happens is the person gets infected with this common cold. Now, the symptoms usually will last for 3 to 7 days, and in common cold we have symptoms like cough, headache, high fever, cold, a sore throat, etc. Two more viral diseases I would like to mention over here. One is chikungunya and the other one is dengue. Both are vector-borne viral diseases. Both these diseases are transmitted by vector, that is a mosquito. Specifically in dengue, it is the Aedes mosquito which is responsible for the transmission and carrying it to a healthy person. Dengue and chikungunya basically will have symptoms like high fever with chills, headache, muscle pain, joint pains, and the treatment basically involves use of painkillers and analgesics like paracetamol, which will be prescribed to the patient until the symptoms subside. The third group of diseases are protozoan diseases. The example of a protozoan disease is malaria. Malaria, the organism which causes malaria is Plasmodium vivax. There is another species of Plasmodium which is Plasmodium falciparum, and the malaria which is caused by Plasmodium falciparum is the most dangerous one. The transmission again here, it is a vector-borne disease transmitted by an infected female Anopheles mosquito. The symptoms include fever with chills, loss of appetite, weakness, etc. Usually malaria is diagnosed with lab tests like the blood tests. The treatment here in malaria is a drug which is commonly used, which is called as hydroxychloroquine. This shows the life cycle of Plasmodium. Let us look into the stages of life cycle of Plasmodium. Here we can see a mosquito which is rather carrying the sporozoites or the malarial parasite. It, when bites a healthy person, releases the sporozoites inside the blood of the infected person, and these sporozoites then reach the liver of the person, wherein the liver cells will help in the multiplication of this protozoan. The liver cells will enable this protozoan to reproduce asexually, and then the progeny is released into the blood of the infected human. The RBCs are also having this protozoa which is going to multiply within this asexually, and within the blood also they are getting circulated, and when the progeny of the protozoa or the malarial parasite increases, usually the human will show these symptoms, that is the fever with chills, loss of appetite, weakness, etc. This is basically the asexual reproduction part within the red blood cells. Within the red blood cells, sometime later the parasite will also reproduce sexually, and the gametes will be formed. The gametocytes which are circulating in the blood of this infected human being. Now when the mosquito, female Anopheles mosquito again bites this infected human being, it is going to take along with the blood this gametocytes, and the remaining part of the life cycle of this malarial parasite is going to happen in the mosquito, where the mosquito's gut is helping in the fusion of the gametes and resulting in the formation of certain other structures. So sporozoites are formed here, where the salivary glands of this mosquito is going to carry the sporozoites, and this mosquito when further on goes and bites another healthy human being is going to release all the sporozoites in that human being, thus infecting that human being with malaria. The second protozoan disease is amoebiasis, which is also called as amoebic dysentery. The causal organism here is Entamoeba histolytica. The target area specifically is the large intestine in humans. The symptoms of this disease includes constipation, abdominal pain and cramps, stools with mucus and blood clots. Usually transmission is by vectors like housefly. So housefly is a carrier here. Usually the infected human being when passes the feces will contain these amoeba or Entamoeba histolytica, and the feces which will contain this traces of the parasite will be taken by the vectors like houseflies, and then if the food is uncovered or the water is not properly stored, then they can be transferring this ant amoeba into this food and drinking water. And when this food and drinking water is taken by the healthy human being, the organism is going to get transferred. So, this is a picture of how under the microscope ant amoeba histolytica would look like, and this is the target area, basically the large intestine of the human. The next set of diseases is diseases caused by helminths. The first disease is Ascariasis. The causal organism here is the Ascaris species. The target area is intestine again. So, Ascaris is an intestinal parasite. The symptoms include internal bleeding, muscular pain, fever, anemia and blockage of intestinal passage. The transmission here also happens through food, water and soil, which is contaminated with the eggs of the parasite, which have come from the feces of the infected human. The next disease which is caused by helminths is filariasis, which is also called as elephantiasis. The causal organism here are the filarial worms, which are basically Wuchereria bancrofti and another species which is Wuchereria malai. The target area in elephantiasis is the lymphatic vessels of the lower limb as well as the genital organs. The transmission here, again elephantiasis is a vector borne disease. So, the female mosquito is responsible for the transmission of filarial worms. The signs and symptoms includes chronic inflammation of lymphatic vessels in the lower limbs and gross deformities in the genital organs. So, here is a picture which shows one of the limb is affected by the filarial worm. So, it has swollen. So, there is inflammation here, and this is not only restricted to the lymphatic vessels of the limbs, but also the genital organs, which will show deformities. The next set of diseases is the fungal diseases. The fungal diseases, first one we will look at the ringworm infection. This is a very common infection, the ringworm infection. The causal organism here is the fungi from varied genera, the Microsporum, Trichophyton and Epidermophyton. The target area basically is the skin, nails and scalp of the humans. The symptoms include appearance of dry scaly lesions which is accompanied by intense itching. Transmission again is from the soil into the nails of the human being, or when the towels or the clothes and combs which are used by the infected person will be used over and over by the other members of the family. So, the disease can be transmitted in such a way. Now, overall we have to remember that we need to prevent and control these infectious diseases. So, some of the general precautions one need to take here are mentioned as some important pointers. Maintain personal and public hygiene. One must remember that one should maintain a personal hygiene where we need to clean our body regularly, clean our nails regularly and cut our nails regularly. Also, as far as the public hygiene is concerned, if we are having a contagious disease like common cold, we need to maintain public hygiene. We need to take precautions so that we do not sneeze directly or cough directly into the mouth of somebody other person. So, we need to take proper precautions. Then we need to also consume clean drinking water. This clean drinking water will mean that the water which is boiled thoroughly and also which is closed and kept. The vegetables and fruit should be properly and thoroughly washed before consuming them, and the food should be properly cooked before consuming it. Then the proper disposal of wastes and excreta should be undertaken so that we don't spread the diseases or we are not the agencies through which the disease is transmitted. And then there should be periodic cleaning and disinfection of water reservoirs, pools, tanks, etc. So in cases where we store water for a longer period of time, we have to take into account that these reservoirs or these tanks are periodically cleaned, so that the vector which are responsible for transmission of various diseases like malaria, chikungunya and dengue do not thrive and do not breed in this stagnant waters. And if at all there are stagnant waters anywhere, they should be properly disinfected and properly cleaned. So these are some of the prevention and control measures that one need to take. We will now come to a group of infectious disease which is called as the AIDS. It is also called as acquired immunodeficiency syndrome. The causal organism in AIDS is basically a virus which is called as the HIV or human immunodeficiency virus. It is belonging to a group of viruses which are known as retroviruses. Usually we have to remember, when the person gets infected with HIV infection, that does not escalate within a short time into AIDS, rather it takes some amount of years, usually a time lag of 5 to 10 years, which is called as the window period, after which the HIV infection becomes AIDS. Let us see the modes of transmission in HIV. The first mode of transmission is sexual contact with the infected person. The second mode is transfusion of contaminated blood and blood products. Thirdly, by sharing infected needles, as in case of intravenous drug abusers. And lastly, from infected mother to her child through the placenta. So people who are generally at the risk of getting AIDS are usually the people who will have multiple sexual partners, the ones who would require continuous blood transfusions. So if the syringes or if the medical equipments are not properly cleaned, they can be responsible for transmission of this HIV infection. Then usually we see that the drug abusers which are using syringes to intravenously take drugs can also be at high risk of taking HIV infection. And then usually the infants or the child which is born to a infected mother can have high chances of getting HIV. One should remember, HIV is only spread through body fluids and it is not spread by physical contact. So if a HIV infected person is around you and has touched you, you are not going to contract the disease. So the social stigma or the taboo associated with people suffering from this infection, HIV infection, should not be encouraged. Let us see the life cycle of HIV. As we know, the HIV is a retrovirus. It has a RNA genome. The RNA genome, when enters into the body of the person, firstly it will go and enter the macrophages. So this is a macrophage cell in which the infection or the virus has taken entry or gained entry. We have to remember, macrophages are the cells of our immune system. So these macrophages, when takes up these RNA, they are going to convert itself into the double-stranded DNA. The RNA genome of the virus is going to convert itself into the double-stranded DNA using an enzyme which is called as reverse transcriptase, and the process is called as reverse transcription. The result of reverse transcription is production of a double-stranded DNA molecule. This DNA molecule is going to incorporate itself or become a part of the genome of the host cell, that is the macrophage cell, and thus it is going to direct the DNA to produce the viral particles. Once the viral particles are produced by the DNA using the host cell's machinery, they are packaged into the mature viral particles which are released from the macrophages, and as soon as they are released from the macrophages, they are going to go and infect the helper T cells. The helper T cells, again the cells of the immune system, and here the helper T cells are also going to usually replicate and produce progeny viruses. So, thus we can see that in the person who is infected with HIV infection, we have the macrophages becoming the HIV factory, and the T helper cells which are present are going to drastically reduce in their number. Because the T helper cells or the T lymphocytes are reducing in number, we can see that the secondary infections that usually would have been prevented are now dominating the or infecting very vigorously this HIV infected person. So, in all, the HIV infected person becomes immunocompromised, and other mycobacterial diseases, other parasitic pathogens and other fungal pathogens are going to easily infect this particular organism, and that is how the HIV is going to transform into AIDS. The diagnosis of this HIV can be done by a test which is called as ELISA. It is enzyme linked immunosorbent assay. The treatment involved here are use of antiretroviral drugs. One must remember there is no cure for AIDS. So, this is partial treatment and it will be not fully effective. So, better is prevention. So, always prevention is better than cure. There are certain organizations in our country and also internationally which are working towards educating people and providing them with proper facilities in order to deal with the HIV infection. Some of the organizations are National AIDS Control Organization, which is called as NACO. There are certain NGOs which are non-governmental organizations working also, and then at the international level we have the WHO, the World Health Organization, which is also disseminating information in various forms and through various media to educate people and provide them with facilities to deal with HIV. We come to the next disease. This is one of the most dreaded diseases because it is responsible for many deaths in the country, and it is cancer. Now, what is cancer? Cancer is a disease which shows abnormal and uncontrolled growth of cells. Usually, it results in the formation of masses of cells which are called as tumors. Normal cells, basically one has to remember, when they grow, there is a property within them which is called as contact inhibition. So, what happens is when normal cells grow, they come in contact with the neighboring cells, and once they come in contact with the neighboring cells, there is a signaling that is happening which helps them to inhibit their growth, and so the uncontrolled growth is checked. This is in the normal cells. Whereas in the cancer cells, one has to remember that this property of contact inhibition is usually lost. Tumors or cancers can be classified into two types. One type is called as the benign tumors and the other type is called as the malignant tumors. Benign tumors are normally the ones which remain confined to their original location and do not spread to other parts of the body. So, there is no spread from the location where they are formed to another region in the body. Whereas malignant tumors are very dangerous because they show a property which is called as metastasis, and this metastasis is nothing but it is going to result in the spread of these cancerous cells from the original site where they are formed to a distant location in the body. That is, when these cells get dislodged from the place where they are originally formed, they will move through the blood, and then wherever they reach, if they find a suitable place, they will get lodged to a newer location and start the cancer at that particular location. So, this is a very dangerous property of the malignant tumors. Let us see what are the causes of cancer. There are certain agents which are responsible for the happening of the cancer, and these agents are called as carcinogens. Carcinogens can be classified as physical, chemical and biological. The physical agents or physical carcinogens could be ionizing radiations like X-rays and gamma rays, while non-ionizing radiations like UV rays. Now, these radiations have the property to damage the DNA, they can penetrate the cell and reach the DNA, thereby causing damage to the DNA. And when the DNA is damaged, it has the ability to transform the normal cell into a neoplastic cancerous cell. The second agents or the second group of carcinogens are the chemical agents. These chemical agents are usually the smoke that is liberated from the tobacco, usually when cigarettes smoke which contain this tobacco, and it is a potent carcinogen as far as the lung cancer is concerned. Asbestos dust also is also responsible for lung cancer. Then we have the polyvinyl chloride and other cadmium oxide and other chemicals which are responsible for different type of cancers in human body. Then coming to the biological agents or the biological carcinogens, we have to remember that there are certain viruses which tend to infect on and off human body, and these viruses have certain set of genes which are called as oncogenes or viral oncogenes. These viral oncogenes, as we know, have the ability to incorporate itself into the human DNA and then bring about change in the human DNA, and then since the DNA is changed, it will result in conversion of the normal cells into neoplastic cancerous cells. Then certain human cells also have these genes which are called as proto-oncogenes, and these proto-oncogenes when get activated can result in formation of cancer. Okay, so these are cells which are present in all the human beings, which is containing these proto-oncogenes, and these oncogenes when activated will result in converting the normal cells into cancerous cells. Let us see how the cancer can be diagnosed and detected. There are various techniques which are used for diagnosis and treatment, diagnosis of the cancer. One is the biopsy and the histopathological studies. In biopsy or histopathological studies, basically the tissue is taken for cytological study. The cell count is analyzed and the other changes are analyzed, which will detect whether the organism's tissue is infected with cancer or not. Then we have radiography, basically which is taking X-rays, CT scans which is computed tomography, MRI scans which is magnetic resonance imaging. So, these are all imaging techniques which will help us to locate the cancers in the internal organs. Then we can also have antibodies formed against cancer specific antigens which can be also used to detect and diagnose cancer. Even techniques of molecular biology can be used for detection and diagnosis of cancer. As far as treatment of cancer is concerned, there are various options which can be studied. One is the surgery, the other is the radiation therapy, where non-ionizing radiations will be used to detect the cancerous cells and they will be destroyed. We have immunotherapy, chemotherapy, wherein different chemical drugs are usually administered so that the cancerous cells can be identified and destroyed. Sometimes combination of some of these can also be used for treatment of cancers. And then there are alpha interferons. There are drugs which contain this molecule. These are cytokines basically called as alpha interferons, which when administered to the cancer patients makes it easy for the cancerous cells to get detected, and thus the treatment becomes easy. Sometimes there are side effects which are related to the chemotherapy, and one we are aware of these. There is hair loss, there is anemia, etc. to the patients who undergo this chemotherapy. Let us now see the details about the immune system of our body. Immunity is generally the ability of the host body to fight the disease causing organism which is conferred to that organism by its immune system. So, the immune system in the human body comprises of the lymphoid organs. These lymphoid organs are the organs where the lymphocytes originate and mature and proliferate. So, lymphoid organs are the places in our body where the lymphocytes or the cells of the immune system will originate, mature and proliferate. Lymphoid organs can be classified into primary lymphoid organs and secondary lymphoid organs. Within primary lymphoid organs, we have the bone marrow, which is the major lymphoid organ, and then we also have thymus. And secondary lymphoid organs, we know that there is spleen, there is lymph nodes, there are tonsils, there is Peyer's patches of small intestine, appendix, and also mucosa associated lymphoid tissue or MALT. This picture shows the different cells or the different organs of the immune system. We have this is the thymus. Inside the bones of the human being, we have red bone marrow, which is basically the primary lymphoid organ. Thymus is also the primary lymphoid organ. We have the spleen, we have the appendix, we have the intestine, Peyer's patches, and then we have lymphatic nodes which are located throughout the body, and these all constitute the lymphoid organs. Let us see what does the bone marrow do. The bone marrow produces lymphocytes and blood cells. Basically these lymphocytes, some of them will mature within the bone marrow, and these will be called as the B cells or the B lymphocytes. Some cells which are produced in the bone marrow will migrate to the thymus, and there they are going to mature into T lymphocytes. When they mature into T lymphocytes, they become antigen sensitive. The B and the T lymphocytes which are present in the primary lymphoid organs are going to migrate to the secondary lymphoid organs via the blood, and this is where in the secondary lymphoid organs where the immune response is basically generated when a foreign particle or the pathogen is encountered. So, the first organ is spleen. It is a large, you can see over here, it is a large bean shaped organ, and it filters the blood by trapping the blood bone microbes. It contains lymphocytes and phagocytes, and it is a large reservoir of erythrocytes. Then we have the lymph nodes. These lymph nodes are solid structures that are located at different parts along the lymphatic system. They trap the microbes and antigens, and when these antigens are trapped here, they are going to be identified by the lymphocytes, and here the lymphocytes activates and thus results in the generation of the immune response. And lastly, we have the mucosa associated lymphoid tissue or MALT, which is basically located within the linings of the respiratory, digestive and the urogenital tract, and it constitutes about 50% of the lymphoid tissue in human body. Immunity can be classified into two different types. The first type is called as the innate immunity, and the second type is called as the acquired immunity. Innate immunity is a very non-specific type of immunity and generally is present at the time of the birth. It will act as a barrier towards the entry of any foreign agents or pathogen into our body. Innate immunities can be of four different types. The first barrier is the physical barrier. Here the skin, the mucous lining of our gastrointestinal tract, respiratory tract and urogenital tract will act as the physical barrier which will trap the microbes. The second physiological barrier, the acid which is present in our stomach, the saliva which is there in our mouth and the tears that come out from the eyes, generally all of these will be again barriers or will prevent the microbial growth. Thirdly, there are certain cells which are called as the cellular barriers in our body. These are leukocytes, specifically named as polymorphonuclear leukocytes or PMNL, neutrophils and monocytes, whereas there are certain lymphocytes also like the natural killer cells and macrophages, which are again the cells of the immune system which are going to destroy the microbes by the action of phagocytosis. Fourthly, there are cytokine barriers. certain virus infected cells will generally secrete proteins called as interferons, and these interferons will protect the non-infected cells from further viral infection. So, this category of interferons are called as cytokine barriers. Acquired immunity is the pathogen specific, whereas it is developed during your life. As and when you are infected with certain organisms, your body develops this immunity and it is called as acquired immunity, and it is characterized by presence of memory. Acquired immunity can be further classified into active acquired memory and passive acquired immunity. The first encounter of the body with the pathogen, when the first the pathogen infects your body, the response or the immune response which is generated is called as the primary response. This response is very slow, and subsequent encounter with the same pathogen will result in a highly intensified response of the immune system, and this is called as the secondary response. In both these type of responses who act, it is the B and the T lymphocytes which are present in our body are going to act upon them and thus generating these responses. It is the B cells which are going to produce certain proteins which are called as antibodies in huge amounts or in large numbers that will be going to react with the antigens, or rather going to react with the foreign particles and destroy them. Whereas T cells are going to help the B cells to produce this antibodies. Here we can see the B and the T lymphocytes, they produce an army of proteins. These are nothing but antibodies which are going to fight against the pathogens. Antibodies are also called as immunoglobulins. So, specifically it is the B lymphocyte which is forming the antibodies, whereas T lymphocytes are going to help the B lymphocytes to produce the antibodies. Here is a structure of an antibody molecule or the immunoglobulin molecule. We can see that each of these antibody molecule has 4 chains, 1, 2, 3, 4. Out of these 4, 2 are heavy chains or longer chains, and the remaining 2 are smaller chains or the light chains. Okay. And then we can see the arrangement is something like this. This is the region where the antigen specifically binds to the antibody. Antigen is the foreign pathogen or any particle of the pathogen that is going to bind with the antibody over here. So, these are the 2 heavy chains and these are the 2 light chains. Generally, there are 5 different classes of antibodies, and these 5 different classes are immunoglobulin G, that is IgG, IgM, IgA, IgD and IgE. Acquired immune response types can be of the 2 types. Since this acquired immune response is generally happening through the blood, because the B lymphocytes are present in the blood, and when the pathogen is present or identified in the blood, the immune response is generated, and since it is happening in the blood, it is called as the humoral immune response. Whereas there is another kind of response which is called as the cell mediated response or the CMI, which is brought about by the T cells specifically. So, the B cells are going to bring about the humoral immune response by the production of antibodies against the specific antigens, whereas T cells are going to bring about the cell mediated immune response where no antibodies are produced, and this cell mediated immune response is generally seen when there is a graft rejection that is happening. We often see that there are certain transplants that are taking place in the human body where there is organ which is non-functional is been replaced by a donor's organ. So, this donor's organ usually needs to be first checked for the tissue matching, and only when the tissue typing and the blood group typing is done, and when this tissue typing and blood typing is done, only then the organ can be transplanted into the body of the human being. The immune system of the human being has the ability to differentiate between the self and the non-self organ, and in order to protect the transplanted organ from being detected by the immune system of the body. There are immunosuppressants which are administered to these patients who have undergone the transplant surgery. Okay. And then usually when the immunosuppressants are not working, or rather they are not given, the cells of the body or the T cells of the body are going to destroy the organ transplanted. So that is called as graft rejection, and that process is brought about by the T cells, and hence graft rejection becomes an example of a cell mediated immune response. Immunity can be also classified as active immunity and passive immunity. Active immunity is when the host body is exposed to a pathogen or antibodies are produced in the host body. This is basically called as active immunity. So generally it is the first hand exposure to the pathogen by the organism's body for response towards which antibodies are generated, and this is called as the active immunity. Active immunity is basically slow and it takes time to be fully effective. The examples of active immunity can be the natural infection process, or it can be deliberate injection of a weakened pathogen, that is during the time of vaccination, active immunity is induced. Whereas passive immunity, if we see, when there is ready-made antibodies which are given directly into the person who is infected with the pathogen, it is going to directly fight with the pathogen, and this kind of immunity that the body gets is called as the passive immunity. Passive immunity is rather a type of immunity when the quick response is needed. Passive immunity can be given by administering antibodies, preformed antibodies directly to the person's body. Example of passive immunity is basically colostrum, which is secreted by the lactating mothers. They contain huge amounts of antibodies which is going to protect the infant, okay, which is feeding on the colostrum. Similarly, if a person is bitten by a snake and if is having a serious tetanus infection, it needs to be administered preformed antibodies like antitoxin and antivenom, which is also an example of passive immunity. Coming to the next part, that is immunization. We have to remember that the body's mechanism to fight against the diseases has to be strengthened, and in order to strengthen the defense mechanism of the host body, usually administration of vaccines and other things have been generally used as a greater medical tool in the world. So inoculation of vaccines generally to prevent the diseases is called as immunization. It is helping in strengthening the defense mechanism of the host. It is a very cost effective and powerful tool of the modern medicine. What are the things that can be used to immunize a body? It can be vaccines, it can be directly the antibodies, and sometimes the anti-sera. These are the agents of immunization. Vaccine. Coming to vaccine, vaccine is an agent of immunization as we know. It is nothing but a preparation of antigenic proteins of a pathogen, or it is an inactivated or a weakened pathogen that is introduced into the body of the organism. Upon vaccination, the immune response will be generated, that is the body is going to identify the vaccine, that is vaccine is nothing but the antigenic protein or a pathogen which is inactivated or weakened. So, the immune system is going to identify that weakened pathogen and it is going to generate an immune response. Upon the generation of immune response, these pathogens will be destroyed. But what it is going to also do is, this is very important, generate memory B and T cells. That means in future, subsequently, if the organism of the human is exposed to the same pathogen, the memory B and T cells are going to immediately recognize the pathogen and produce massive amounts of antibodies, and these antibodies are then going to destroy the pathogen. So, this is the advantage of vaccination. Different vaccines that have been very successful are smallpox, oral polio vaccine, we have the BCG, rubella vaccine, hepatitis B, so on and so forth. As I told you before, passive immunization is also a type of immunity which is when a quick immune response is required. Like in case of tetanus and snake bites, we cannot wait for too long until the body is generating the antibodies. So preformed antibodies are administered to these patients. So preformed antibodies will basically mean an antitoxin. It is an antibody towards the toxin generated by the tetanus pathogen, and anti-venom. It is an anti-venom or anti-venin which is basically acting against the venom of the snake, and these are going to be administered to the patient, and thus the patient will get the advantage of the direct administration of these antibodies, and this is called as passive immunization. Let us see about allergy now. So, when the immune response generally shows an exaggerated response to certain antigens which are present in the environment, and this is called as allergy. We often see that whenever we go out in a cold morning, we usually have sneezing and watery eyes, a running nose, or difficulty in breathing. This is because these are all the symptoms which are associated with the allergy. So, there are many allergens, or allergens are nothing but these are the substances which produce allergic response are present in the environment. So, these are going to produce all these allergy symptoms, like we have rash formation, runny nose, itching, red eyes, and tears may also come out, that is lacrimation. So, these are symptoms of the allergy. Now, why are these symptoms generated? These are generated because there are certain cells in our immune system which are called as mast cells. They are type of the white blood cells. They will release chemicals like histamine and serotonin which are going to produce this exaggerated symptoms. Usually the antibodies that are produced against this type of antigens are IgE type. As we all know that substances which are going to generate this allergic response are called as allergens. Some examples of allergens are mites that are present in the dust, pollens, animal dander, which is nothing but the hairs of the domesticated cats and dogs and other animals. So, these are the allergens which are present. Usually allergy can be diagnosed by means of a patch test where smaller doses of the antigen or the allergen is administered to see which one is the exact cause of your allergy. Then treatment is basically here antihistamines, that is the histamines that are secreted by the mast cells to antagonize or neutralize the effect of histamine. Antihistamines can be administered. There are adrenaline also which is given, and sometimes steroids are also given to treat allergy or manage allergy. And the last part of the immunity is autoimmunity. Generally we have to know that the immune system, when fails to recognize the self and the non-self cells and attacks its own body cells and damages the healthy tissues and organs of its own, this kind of immune response or this kind of an aberrant or abnormal immune response is called as autoimmunity. And the disease which is generally happening due to this aberrant kind of behavior of the cells of the immune system, it is called as autoimmune disease. And the example of autoimmune disease is rheumatoid arthritis. The topics related to drug and alcohol abuse will be dealt separately in the next part of this chapter.
Prudent Scholars, powered by Lupin Pharmaceuticals.
अब आपकी सुरक्षा आपके हाथों में। ल्यूपी सेफ हैंड सैनिटाइजर, आपका और चेन्नई सुपर किंग्स का फर्स्ट लाइन ऑफ डिफेंस। अब आपकी सुरक्षा आपके हाथों में। ल्यूपी सेफ हैंड सैनिटाइजर, आपका और चेन्नई सुपर किंग्स का फर्स्ट लाइन ऑफ डिफेंस। अब आपकी सुरक्षा आपके हाथों में। ल्यूपी सेफ हैंड सैनिटाइजर, आपका और चेन्नई सुपर किंग्स का फर्स्ट लाइन ऑफ डिफेंस।